So good morning, one and all, and thank you so much for joining one day international program on international conference on applied nanotechnology. So myself, Dr. Ashutosh Kumar Shukla, and I'm the founder and director of Drug Chemistry Research Center. So Drug Chemistry Research Center is powered by Nai Suba NGO and established in 2020 at Indore, Madhya Pradesh, and nurtured and promoted under the special guidance from the persons of cheminformatics and bioinformatics and other related fields. So DCRC is basically dealing with the in the field of in silico drug design and discovery in the central region, India, and organizing various conference, national, international webinars and training programs to the participants or the clients related to the Drug Chemistry Research Center for the enrichment of the knowledge. So, why in silico research work is required? Like the PETA and other ethical committee, animal ethical committee has issues that it's not good to killing animals. So it's better to repurpose the drugs before going for the in vitro or in vivo or animal testings. So basically, we are dealing with drug repurposing. So first, we have a lot of drugs like in many lakhs. So first, we go for the traditional drug discovery with high risk and low return. So after the drug repurposing, we can go for the computational methods and a literature survey, and there are different type of screening such as phenotypic screening on the basis of cell, disease, and animal, and different type of methods like network pharmacology and other in silico method to repurpose the drugs. So after the repurposing, we will finalize the drug for the in vitro in vivo activities. So, Drug Chemistry Research Center is dealing in various fields of sciences like network analysis, transcription signature, protein comparison, binding site detection, docking, and ligand similarity, machine learning, data mining, artificial intelligence, and we are also running a CRO, contract research organization, and we have various collaborations where we are performing the research. And now Drug Chemistry Research Center is DCRC is also involved in various research areas like sequence level DNA and RNA protein related work, interactome and organelles, cell signaling tissues. So by using these methods, these materials and methods, we go for repurposing, finalize some compounds for like which are good candidates and having a good targeting properties, and after that the cellular response, animal models, and electronic health record and clinical trials. So these are the, you can say, the pathway or finalizing a drug after the repurposing or various using various in silico methods. So beside this, we are also doing some collaborative work like in vitro cell line based activities for cancer, diabetes, and arthritis and other related diseases. We are also performing some antimicrobial activities for bacteria and fungi. And beside this, we are also doing some design, scheme designing and synthesis of molecules. And we also doing a plant extraction isolation of constraints from the plant. After that, we are also involved to do the characterization from the various modern techniques like HPLC, IR, LCMS, GCMS, NMR, XRD, and different other modern techniques. So we also sign MOUs from different institutes or universities, and some software developing companies, and some production companies like Game Master Laboratories, and we have collaboration with PBRI, SIRT College, Bhopal, LNCT University Bhopal, and Mansrovo Global University, Venus Institute of Pharmacy, Sagar Group of Institution, and Mittal College of Institutions, Truba Group of Institutions, Chameli Devi Institutions, Indore, Oriental Group of Institution, Bhopal, and NRI Group of Institutions, and many more institute and universities going to sign MOU with us in the coming months. So these are our MOU bodies. Unicode Software Private Limited is a Wilmington and USA based company, which is also have a subsidiary body at Bhopal, Madhya Pradesh. They are developing softwares for dcrc labs. We have various collaborations from different countries like US, France, Canada, Egypt, Russia, and Australia and South Korea. We are working and completing their projects in collaborations. We are also collaborating with various bodies in India like CSIR labs, IITs, NITs, various government and private universities and companies. Further, we have conducted various activities like more than 11 national international programs, training programs, and conducted various complete projects for bachelor's, master's, and PhDs. We have more than 960 or 1000, you can say, participants from DCRC labs who have enriched with the knowledge of in silico and other related research areas. And we have also published various research and review articles and book chapters, and some are under review. And so this one, the latest latest, or you can say the highest impact factor journal publication from the DCRC labs, published in Biomedicine Pharmacotherapy. Publisher was the elsewhere. And we beside this, we have various contributions and collaborative work from different institutions like CSIR and IITs and central universities. So we have published various research review and book chapters. So this is our contacting or you can mailing address. So you can contact us for further research or contributions. So I just want to let you know today's speaker, Dr. Ashwani Mishra, sir, and so Dr. Ashwani Kumar Mishra sir is a professor at the Department of Pharmacy, Barkatullah University, and he has completed his master's M Pharma in School of Pharmaceutical Sciences from RGPV Bhopal, and PhD from Barkatullah University. Sir has more than 15 years of experience and 15 years experience, and he guided more than 70 PGs and more than 95 B Pharma major projects. Sir has published various research and review articles in reputed journals, more than 100 research and review articles. He has also published one book and two book chapters. He also conducted some schemes like communication and popularization of science and technology, NCSTC, from National Council of Science and Technology Communication in 2013. He was awarded as a Young Researcher Award in 2021. And he has more than like he has many years of experience, and he is also members of more than 40 committees and boards, including a member of board of studies, Department of Pharmacy, Barkatullah University, students coordinator in drug disposal awareness under FHER New Delhi. under the Niti Aayog, and he is also member of a Society of Pharmaceutical Education and Research. Sir has various of experience as a team member, and he is in research in various scientific bodies. He is also life member of APTI and team manager of Madhya Pradesh Yuva Utsav 2017. Sir was also jury member in 27th National Children's Science Congress, and he is also jury member in IPA Davinder Pal National Elocution Competition in 2019 at Madhya Pradesh. He was also ambassador of Drug Discovery Hackathon 2020 in the first phase, and conducted as a joint initiative of AICT, CSIR, and supported by Principal Scientific Advisory, Government of India. So we welcome Dr. Ashwani Kumar Mishra sir for giving a lecture on applied nanotechnology, and we welcome you, sir. And we are waiting for sharing your experience with us and DCRC labs. Thank you so much, sir. Now over to you, sir.
Okay, thank you very much, Dr. Ashutosh Shukla. It was such, I should say, huge introduction for a small person like me. And the journey of DCRC, as you have shown, it has a number of avenues for the researcher if they understand the importance of such a lab in Madhya Pradesh, which is actually a matter of, I should say, pride for the researcher in Madhya Pradesh, not only in MP, in India. So I should people should come and do the work with you in the collaboration, because whatever avenues have been shown by you in the first presentation slide, they are remarkably good, and they are current trained research technology. So it's really a pleasure for me that I have been called here for giving this lecture, and I really thank you to the director and the whole team of DCRC Lab for this invitation and considering me for this opportunity. So now I'm going to share the slide.
Sure, sure. Thank you so much, sir. Can we have your video?
Yeah, sure.
Yeah.
Uh, is it visible?
Yes.
Okay. So should I? Okay. Is it in presentation mode now, the slide, or it's there in the open mode only?
Not now, sir.
Okay, I think it is going to be. Is it
Yes.
okay now?
Yes.
Okay. So as we all know that mm, nanotechnology, which is considered as a most fine, most refined, or even I should say the technology of current age, has been doing tremendous task in human health, not only in terms of providing the therapeutic effects and the therapeutic benefits, but even it has shown its remarkable uh, futuristic application in diagnostic also. So the conference, which is based on recent techniques and even the theranostic approaches, where these kind of potent things will be discussed. My topic of today's lecture is like theranostic, a heterogeneous approach of nanotechnology in salubrious metamorphosis of human health. So before proceeding ahead with this topic, I would like to tell you that this particular term theranostic is actually a combination of two words. One is therapeutic application of nanotechnology, and the other one is a diagnostic application of a nanotechnology. Before continuing it, I do have a senior with me, Professor Nilima Goswami, ma'am. She is not only senior, even sometimes my mentor also in many conditions. So, a very good morning, ma'am, and sorry to interfere in between.
Good morning, Ashwani.
Okay, ma'am. So now today, as the topic is vital, and we do have very good, I should say, knowledge reservoirs with us. So it's even a pleasure for me to exhibit something in front of all of them. So in theranostic as it is a combination of two therapeutic as well as diagnostic avenues of nanotechnology. My first emphasis will be on the diagnostic approach of nanotechnology, in which we will talk about how the mechanism has been utilized in the world by using these nanostructure stuff for, I should say, the diagnosis of human disease and for the welfare of human disease. So, wait a second. Okay, so the before moving ahead, one thing which I would really want that we Indians should know, that this particular term theranostic has been coined by this John Fletcher, who it was in 1998, and we people have started listening about it probably in the last decade. So we have to understand that how far and how speedily the science is actually moving salubriously in Western world as compared to India, and that's how we have to have a mode in which we can increase the speed of scientific research even in India, so that very soon a term will be coined by an Indian scientist and will be elaborated definitely in Western world. So I'm looking forward for the learners who will be there today, and they will definitely fulfill this notion which I have to move ahead with the country and do well in this field. So when it comes to theranostic, the diagnostic start with, like, as we all know, that it can be MRI, it can be NIS, can be PET, SPET, or probably NIR fluorescence, where the nanotechnology is booming actually. And when it comes to therapeutics, where we all know that there are lots of drugs which can be utilized in the treatment of patients. Even if you started counting the name of disease, the nanotechnology is really doing miraculous, I should say, application in that specific disease treatment. So it can be photodynamic, chemotherapy, gene therapy, hypothermia, radiation. Even if you go for the general disease, then also the nanotechnology is becoming a promising mode of giving therapy and increasing the viability of the formulations by its own specific features and attributes. Now, as like we know that there was an era when we used to talk about macros. Then the era came where we started talking about micros, and now the era which is nano. And I'm not very much sure, but yeah, one day we will be having one more column here, which will be even much more advanced. So today, the era which we people are enjoying, having the facility, research facility, and even the dimension of nano in order to conduct our research and providing the therapeutic salubrious benefit to the patient in this consistently changing world. Now, how nanotechnology is being used in the diagnostic of human disease? Through this slide, I'm trying to give a gist of this particular thing. So here you can see human body, which is actually a miracle of the God, having lots of chemicals, having lots of protein, ligand, multi-component system is there, multi-compartment system is there. And in this particular body, you may see there are lots of chemicals which is being secreted every second. And the kind of channelization, the kind of I should say, uh, effect and response which is being exhibited by body is is what do we call it? We are today. So right now, even if I'm talking, there are lots of chemical, reactions which are going in human body. You may call it any science. It can be biochemistry, it can be genetics, it can be pharmacy, or it can be even, I should say, the genome which is being showing its perfect function in human body. So whenever it comes to utilization of the nanotechnology in the diagnostic, the conventional diagnostic method is based upon the symptoms of the patient. Like suppose if someone is having headache, so the patient will ask or will ask to the doctor that I'm having these kind of problems, so what could be the solution of it? Or probably someone has got acne, then a sign of a polyp on the skin will appear. Or probably someone has got cancer, probably it's a breast cancer, then the nodules are there, or the swelled alveoli are there, which are actually started feeling by touching, or probably it could be a kind of physical or phenotypic sign which actually make you to be aware of disease, and then we rush to the physician, and then the disease is being diagnosed by using different diagnostic technique available in the country, and finally disease is identified. What nanotechnology is doing in this particular case, that we, the people who are considered as a homo sapiens, a social person, having body where lots of chemical signals are being secreted in the body. It can be the discharge of cell, it can be a discharge of gland, it can be certain chemical which is exhibited by the blue dots in this particular photograph. In these blue chemicals or even the fluids are being secreted continuously by a healthy cell, whether it is a sign of the healthy cell, or probably it's a sign of an micro environment or a nano environment where a cell is excreted or even releasing certain chemical which shows its vitality or probably the better health status. The secretion may be in the form of certain DNA, it can be certain form of protein. These signals are being consistently secreted if the cell is healthy. Once on the other picture, you can see the perforations on the cell membrane, the some kind of yellow particles which are actually secreting out from the cells. Once the cell started becoming, I should say, diseased, some other kind of chemicals are being secreted by the cell. So these particular chemicals or biological fluids are considered as a biomarker in scientific community. These biomarkers which are being secreted by the disease cell is actually some kind of genetic material, or it can be the protein biomarkers. So some of them are genetic biomarkers, some of them are protein biomarkers. In nanotechnology based diagnostic technique, these biomarkers are being identified, and on the basis of the identification of these biomarkers, a disease is being first screened. On the basis of the screening of disease and different techniques which are being involving in the process of selection of the specific sample, and then probably amplification of the signal, and then detection of the disease is being conducted. Once it will be done, definitely the identification of disease will be even much more earlier than the sign of the physical or phenotypic symptoms come, because the disease is being diagnosed at a nanomolecular level where only the chemicals or the body fluid uh, or even the fluid which are actually being analyzed. So it will take a time to go through, or probably showing the phenotypic characteristic once the enough amount of the fluid or probably these kind of chemicals being secreted by the body. So this technology is why far better, because like the sign of the physical illness appear on human body through cell level only. The nanotechnology based diagnostic technique are trying to find the disease, and some of them are 99% correct as per the report which is being generated by a very good company, US based company, that is nanopure. So this is how the nanotechnology is actually being used in the screening of the disease and probably for the diagnostic of the disease. And this is what do we call it, the science of 21st century. Now, this is the cell as we people probably, let me move ahead. Okay. So this is particular, the first photo is showing a kind of well, where in this well, some nanoparticulate based bioligand are inserted with the body fluid. So what is it actually? Look at the second picture. Nanoparticles under 100 nanometer structure shows these kind of perfectly red. It is gold nanoparticle. First of all, it is not a polymeric nanoparticle. So these gold nanoparticle under 100 nanometric range shows these kind of perfect red color. Once they are collected in enough amount, they shows true red color which is actually kind of appear like a dye. These red nanoparticles are actually uh, subjected through surface engineering technique where biological ligands are being attached. So these horn-like structures which you are observing in this second picture are actually nothing but these are the biological ligands. These biological ligands have a tendency to bind with the receptor, and the chemo secretory material which is actually secreted by the cells. If the chemical material or the biomarker which is being secreted by the cell is bound with this biological ligand, and it shows the response probably in the kind in the mode of fluorescence, or it shows the response by accumulating in a specific site of the cell. Once these kind of accumulations are being observed because of the amplifications of the signal, these signal are being detected by the different kind of scanners, and the disease is being confirmed. So what does nanotechnological diagnostic technique do is this: from these kind of chips which are being inserted into the different sophisticated machine, which carry these kind of 100 less than 100 nanometer gold particle, which are being articulated and designed in such a way with the biological ligand, so that once a typical kind of chemical is being secreted by the cell, these signals being amplified, and ultimately it is screened, diagnosed, and finally disease is being confirmed. So this is what do we call it, a process of 2 to 3 hours. But if we talk about the conventional technique of determining disease, which even take, like you can see in this figure, it's like a culture plate technique which is widely used in Indian diagnostic purpose. Apart from it, there are more, so I'm not talking about only one thing. And so the report which actually come out from culturing, then identification, and even the susceptibility test of that particular kind of antibiotics, it takes 10 to 14 days. So what miracle this nanotechnological diagnostic technique is doing is that it is minimizing the time of diagnosis. It is giving the assurance because it is based on certain chemical which is being secreted by human body. And the other thing is the chances of variability, and even I should say the variation is less, because the process is a single pipeline approach where the incorporations of the variable factors are less as compared to even the culturing technique where we have to use different kind of culture media. So the question of the purity is raised, the question of the growth of the microbe is raised, the purity of the microcolony which is being growing on the plate is a bit questionable stuff. Even the process of determining whether you go for the zone of imbibition technique, or even you may proceed by some other mode of technique where we are making a well or this diffusion method. There are multiple modes. Even if we go for organoid technique, which is even considered as a multifold organoid model for the culture technique, is definitely a bit doubtful, because the incorporation of various materials are there, various techniques are there, and even the sterilization is definitely a huge problem if we talk about these kind of microbial or microbiology based techniques. So if you are moving ahead with these kind of techniques, so you can easily compare the time duration which is required for the diagnostic of human disease based on these two different techniques. So that's how the topic of this particular lecture is theranostic, because whenever we move and walk with the wind, we definitely talk about the latest technology which even some of us can implement it in our own country and definitely do miracle by utilizing it for helping the patient, which is actually the soul of the function of a pharmacist or even true researcher who is working in the field of health science. Now, these are the structured material which are actually being used in the diagnosis, and they are nanostructured material. It can be starting from fluorines, carbon nanotubes, and they all are in nanometer scale. And these fluorines and carbon nanotubes are being used as a nanowire and a biosensor for diagnostic. I have already mentioned you the importance of gold nanoparticle. These are their size. The nature is spherical. There's electronic and optical and thermal property they are showing, and they are again used in the diagnostic and detection of the biological molecule at the low concentrations. So as being a pharmacist, we all know that what is the importance of the concentration, as long as the therapeutic effect as well as the therapeutic response is concerned. Now the quantum dots, which can be 1 to 10 nanometer as it is specified, again is spherical. these cadmium and zinc selenites, which are being used in vitro diagnostic imaging technique. So it is not that this nanoscience or the nanotechnology is actually a kind of hypothetical world. It is a world which is being adopted by the scientific community, and right now it is a technique of today. It is not a technique of future. So we have to consider it, and that's how we can do miracle. Now this is the first application. So I have only used some example, as the time limitations are there, and every such example is easy to speak, but there are if and buts, do's and don'ts, and pros and cons in everything. Similarly, in this also, which is one of the technique of using nanotechnology on the diagnostic basis, is silicon nanowire technology. Now, what does it do? See, the base plate, which you can see down here in this particular snap or probably picture, where you can see there are two orifices. From one orifice, the fluid is inlet into the sample holder, and the other orifice is having fluid which is coming from the outside, which is indicated by the two specific arrows. Once the fluid is inlet, it is a body fluid which has to be proceed through the inlet system. These silicon nanowires are actually the nanostructure system which actually attach the biomarker, which is actually the biological ligand, or probably uh, a specific ligand which is secreted by human body in certain disease condition. So once this particular wire is being exhibited in front of those biomarkers, these nanowire are trying to bind with the specific biomarker. And once these silicon nanowire biosensors actually analyze for that kind of biological marker, and it detect that what kind of biomarker is being attached with such a silicon nanowire, then the specific disease can easily be identified by this technique. So there at the bottom, the three cases which have been exhibited, that prostate specific antigen is surely being analyzed by these technique. It is used in Western world as a promising technique for it. This carcinoembryonic antigen, which is actually being used in this technique, and it is again repetitively used in Western world, and similarly mucin binding one. So these are the methods which are actually being utilized in experimental work for utilizing as a diagnostic mode by using nanotechnology. So next is cantilever and its application in the diagnostic. So these cantilevers are nothing but, you can see there is a tip of the finger. It is index finger where the tip actually is the cantilever is there, which is indicated by the arrow. So these cantilever is actually shows the deflection and resonance frequency depending upon the sensitivity modulated by the binding of the surface immobilized reagent to the biomarker protein of the nucleic acid. So, like as we can understand simply by considering that whenever there is a particle or probably a nanostructured particle, and you are trying to stick the particle with some more particle, on the basis of the frequency which is being exhibited by the particle will definitely be hampered because of the presence of some other material around it. So the variation into the frequency which is actually being detected in this technique, because the more particle you accumulate, and these particular nanocantilevers are actually specific for the binding with certain kind of biomarkers which are secreted by the human body in certain disease. Like at the bottom you may see the example which is quoted by this particular researcher, and it is used in the inflammatory bowel disease. So the secretions of the human cell, which is actually being secreted at the time of inflammation, are being detected by these nanocantilevers. And if depending upon the frequency which is being exhibited by the cantilevers, we actually trying to understand that, okay, that the inflammation is there. If the signal are, the frequency are being resonated or even modify majorly, then it shows that the the biomarkers which are being secreted are in high number. It shows that this inflammation is not a minor inflammation, it's a case of severe inflammation. So normally in India, what does doctor do? That they give you the non-steroidal anti-inflammatory agent or probably anti-inflammatory drug unknowingly. You, if the inflammation is, it's something what do we call it, a modified mode of inflammation. I should say, like, it's not severe inflammation, it's like a minor inflammation, then also the dose is common. And even if the inflammatory response is severe, then also the dose is common, though dosing frequency can be varied. So in this particular way of determining the inflammation, if you are finding that the biomarker concentration is less, which is actually being adhered to the nanocantilever, so you may even modify the dose, and you can provide the safe and effective therapy to the patient depending upon the response which is being generated by the body in the form of inflammation. So nanotechnology not only do the diagnosis, but it can even harness the process of, I should say, modifying the concentration. So whenever the inflammation is high, you may use high concentration of the drug. Whenever the concentration is less, depending upon the biomarker which are being secreted, you can minimize the dose, even the dosing frequency which is required. And that's how you can give long life to the body, because there will be less drug, there will be low dose frequency or less drug frequency, and there will be less side effects. So this is what do we call it, a smart approach of using nanotechnology in the diagnostic of disease. Let's come to the quantum dots in the diagnostic application. So probably I do not know much about. When I was into my MPharm studies, I came to know about this word quantum dots. And seriously speaking, probably, it's something like you're listening to an alien name somewhere. So it was kind of that stuff for me. And then later on, I myself started reading about it. Then I came to know that, okay, this is something which is a nanostructured formulation, which are used in multiple application, in as long as the pharmaceutical or therapeutic benefits of concern. So here, actually, these are considered the semiconductor, which are actually 2 to 10 nanometer range, and they display the energy level depending upon the size. So why the QDs are used in the diagnostic purpose? First, because of their small size. So what happened, these quantum dots are easily penetrated by the skin, or probably the layer where there is a consistently growth of the cell, which is actually the sign of tumor. And these are, since the accumulation of the quantum dot is specific to the specific tumor cell, depending upon the site of the accumulation of the quantum dot in human body, we can easily diagnose that from where the cancer cells are present, depending upon the concentration of these high amount of quantum dots on that specific site. And through xenographing system, we can xenographing models, we can actually determine the concentration of these quantum dots which are being accumulated at that site, and they are perfectly used in the case of prostate cancer. And even not only in the cancer, but even in the cancer line, it has been studied that the perfect accumulation of these modified quantum dots for the determination and the diagnostic of the cancer in that case. So the next example is nanostructure based MRI MRI contrast agent. So we all know that through the first semester of B pharmacy, for the student specifically, we have been studied about a contrast agent. It can be barium sulfate, which we have studied in the first semester in B pharmacy. So here, these agents are being used in MRI, which is actually a very fine technology of using or probably the diagnosing. So this nanostructure is used to modify the conventional contrast agent, and which is known as gadolinium, which is actually used conventionally. So these nanostructured iron oxide are actually used along with it, and it enhances the diagnostic power of the clinical imaging. Not only it, but it also improve the observations which are being exhibited by this MRI imaging. So the more efficient technique we use, the better diagnosis we can found, and the best treatment the doctor can advise to the patient. So these nanostructure based MRI contrast media are actually again a very fine application of these nanostructured diagnostic avenues, which is actually utilizing in present century, and we are getting the medical benefit by from it. The next example is of the endoscopy of NIRF using quantum dots. So this NIRF is nothing but a near infrared fluorescence imaging technique, which is widely used for the diagnosis of the disease. So what does nanotechnology do over here is that nanotechnology actually enhances the use of certain fluorescence probe. And why again quantum dots? Because of their high penetration and the accumulation into the specific site. So what does they do? They actually accumulate there where there are diseases, and like as in the picture you can see through these small green dots. So it shows that how, like, probably for if we talk about malignant cell, so there are benign tumor which is actually site specific one, and once it started spreading, then the fluorescence which is being exhibited by these quantum dots, it can easily imagine the spreading of these kind of cancerous cell by the process of this near infrared fluorescence imaging technique by using quantum dots, which actually act as a fluorescence probe over here. So you can easily find out by the uh, fluorescence which is being exhibited by these quantum dots, that where actually the specific cell has been spreaded, and whether through what technique the therapeutic effect can be provided. So if it is benign one, then it's easy to treat, because even the operational procedures are quite fine in that case. But once it turn into the malignant tumor, then definitely the condition become more worse. So before proceeding ahead, if these kind of imaging agents have been utilized very nicely by tuning the quantum dots, then definitely the diagnosis will be even much more fine. And as we all know, being an Indian, that most of the time in the the therapeutic benefit which we get, if the diagnosis get poor, then definitely patient will not get the proper drug. And because of that, even if it is a small burning in the heart, become the cause of heart attack, and patient take it, it's nothing but a small burning in the heart. And sometimes people, they drink even eno without considering the severity of such kind of symptoms in human body. So diagnosis, which is again a very important, I should say, the mode, and if it is being done with proper efficiency, with proper understanding, and with proper approach, then definitely, I should say, the proper treatment can be given to the patient. So the next one, I actually this particular side is being shown not to show that the photo of this particular researcher, but to show you people that in 1959, a professor from foreign origin has actually, and he was a physicist who has actually remarkably, uh, I should say, exhibited the name of these nanotechnology. And even from that point of time, it is being into the culture in Western society, and we people have started using it probably from 1 to 2 decades back. So the term nanotechnology had been even coined by this Norio Taguchi in 1974, and this Richard Freeman, who was a professor, invented the scanning Teller microscope in 1981 to discover fluorine, which actually we have discussed before a couple of minutes in a slide. So now let's talk about since it is a theranostic approach, so let's talk about the therapeutic approach of nanotechnology. Though, like, I know that after me, there are great speakers who will definitely move with the depth of this particular thing. So these are only some examples. My only intention is that the student should know that this theranostic mode is actually having the therapeutic avenue also. So these carbon nanotube, which is again example of nanotechnology, definitely, because they are nanostructured tubes, and these tubes are having very good penetration power, and they are definitely used in much more wider way in the treatment of cancer because of their structure and because of their drug carrying capacity. So these carbon nanotubes are, these are allotropes of the carbon, and as it is tube, so they are cylindrical in shape, and they are used as a carrier, because you know what happened, that body is nothing but a multi-compartment model where the reachability of a formulation is really an important parameter. And once the reachability is concerned, a formulation has to be reached from the site of application toward the site where actually there is the enough concentration of does require. And these carbon carriers are actually a finest carrier because of their small size, better movability, surface engineering characteristic, even the modification in the release profile by the application of different polymeric formulation, or even I should say that modification into the I should say the surface property by arranging a specific molecule which will be having affinity to the specific site of human body. We can even use nanotechnology as a booming carrier system to target a specific uh, formulation, having the drug to the specific site where the response is definitely needed. So this again one of the slide where these nanotechnological based carrier is being discussed, in which a nano size carrier is being utilized for the treatment of tumor that is being used in the chemotherapy. There are certain nanorobots which can be utilized to remove the blockage if it's there in the arteries. And nowadays, you know, we normally hear, which is very common in India, that okay, someone has died in the heart attack last night, it happened in someone's life. So we become, if I'm not wrong, we become very much health conscious nowadays. Whatever we take, we even started counting of the calories, whether it's good for us or not. Okay, so the next slide talks about again the two kind of nano formulation. One is nanoparticle formulation, which is derived from the word nano, which means extremely small. So the intention is only to emphasis on the other side of this nanotechnology where the drug is being delivered by the nanoparticle. Then this is the definition where the 10 to 1000 nanometer is the size of these nano carriers, which are actually smart to carry the drug in human body. The nature of these particles are, they are non-toxic, they are biodegradable, and they are sterilizable because they carry the drug in human blood. And whatever we carry in the blood, it should be sterilizable, and that is again the property of it. Normally there are two, which is like nanosphere, which are actually kind of metric system. So these are like physically and uniform kind of disperse, and the nanocapsule, which are having the cavity which is surrounded by the polymer where the drug is being inserted. And these small nanostructured carrier will deliver the drug in human body. There are certain advantages of these nanostructured drug delivery system. So they are site specific. It helps in the maximum therapeutic responses. It may go with the active and passive drug targeting. These are administered by the parental, oral, nasal, ocular route. So you start counting the route. These technique, which is I should say the smart technique, will be able to deliver the drug in that specific cavity as per the modifications can be done in order to reach this formulation into the specific site. So by attaching the ligand on their surface, which is known as a surface engineering technique, where the targeting can be done by using these kind of technology. At the end, without taking your too much time, I have collected this slide by this resource which is mentioned at the bottom. So the application of this medical nanotechnology, which across the variety of area, which may starting from drug, it may starting from therapeutic effect. It is even, as I mentioned earlier in some of the slide, where it is very smartly used and through the sophisticated mode in the diagnostic mode. even in the surgeries and the medical robots, what nanorobots can do in the removal of the deposit which is there in the arteries by using nanorobots. And in general, the sake of increasing knowledge of the human body, it is definitely required. So the more we work on this field, the more we imagine it, the more we understand it, and how smartly we can proceeding ahead by utilizing the boon of science, which is known as nanotechnology. So before moving ahead, actually, I'm really thankful to the Professor Ragini Gotharwal, head of Department of Pharmacy. She allowed me to continue this and to, move ahead in this direction. These people who are there in the list have definitely helped me to move ahead in the field of pharmaceutical research, whether it's Dr. Narhari Shastri, Director CSIR, Dr. Sudagar, he is a assistant professor in Nipur, Guwahati, Professor Dulal Panda, he is again from Nipur. C. Karthikeyan, sir, he is a professor at IJNTU, one of my motivator, one of the person who inspired me a lot, Professor V. Masan. He is the professor from Amravati University. He is doing really well in the in silico research field. He is the one who is not only doing the research in India, but he is being called in various countries for enlightening the researcher how to move ahead in the field of in silico technique. Professor G. Pillai, he is the scientist and even the, I should say, the honor of SAR, machine learning, drug discovery tools, and there are various software which is actually being invented by these scientists, and he is actually doing really well in India, not only in silico drug designing, but definitely by using AI and machine learning models, and their actual empirical application in different institutes of India for moving ahead with the wind of research and giving confidence to number of researchers like me. So at the end, normally it is a fashion that people should speak of using great authors quote, but it is not because it's fashion. I do have respect for these great researchers of India, Professor APJ Abdul Kalam, and one of his quote, which actually has intense meaning, that is, look at the sky, we are not alone. The whole universe is friendly to us and conspire only to give the best to those who dream and work. And that is why probably we are there, because we have been getting help, good vibes, and I should say support from each and everything in this universe. And that is why we are surviving and doing great in the field of pharmaceutical sciences. Thank you very much for your tolerate listening, to tolerate me for such a long time. And at the end, I would definitely thank to Dr. Ashutosh Kumar Shukla, the founder, the CEO, even the torch bearer, I think in Madhya Pradesh, who is actually blowing the wind of in silico research. And we are really lucky that this gem is actually the alumni of Department of Pharmacy also. So I am actually
Thank you so
uh, praising him, not only because he is a part and parcel of the university, but also that the kind of work he has been doing in the field, the kind of length and connection he has been developed in such a age, and definitely we people will be benefited because of his flight and the wings he has, which will definitely take him ahead in the field of research and development, in not only in pharmacy but definitely in multiple sciences. Thank you very much for giving this opportunity to me. Now I'm handing over this platform to you. Thank you very much.
Thank you so much, sir. And hopefully the lecture was very knowledgeable for our towards our participants. And the way you presented the nanotechnology and applied nanotechnology for the application of therapeutics, it was very nice. And so the nanotech is revolutionizing in healthcare via different targeted drug delivery systems and early phased drug discovery and early detection of different diseases like cancer and others. So thank you so much for joining us. I would like to welcome Dr. Nilima Goswami ma'am to for vote of thanks.
Welcome.
And good morning, ma'am. Ma'am, please give a vote of thanks to Ashwani sir. Ma'am, you are, yeah, yes,
Ah,
ma'am.
thank you, Shukla ji.
Thank you, ma'am. Ma'am, we welcome you to for vote of thanks to Professor Dr. Ashwani Mishra sir.
Okay.
Yes, ma'am. So I would like to welcome Dr. Harshita Tiwari for vote of thanks and her query related to applied nanotechnology.
Thank you very much, sir, for giving me this opportunity. So I would, from the behalf of whole dcrc team, I would like to thank Dr. Ashwini Kumar Mishra, sir. And as a naive, I am purely from the computational chemistry. I understood the talk very well. So I totally means hope that participants may also have learned from you. And it was really very insightful, and it has instilled us with great knowledge. So thank you once again, sir. So I have just a little query. So when we talk about drug discovery, so we generally talks about the admit properties of the compounds, which are the major hurdle for their approval during late phase of the discovery. So is it possible with the nanotechnology that we can reduce the particle size and it might positively affect the PKPD of those compounds?
Well, definitely it is possible because whenever the PKPD pharmacokinetic and pharmacodynamic is being concerned, as we know that even by making certain structure 10 nanometer less, we can modify not only the surface property but also the attributes which are associated with that kind of particle. So when it's come to pharmacokinetic and we consider the small particle, then human body, which is considered as a multi-compartment model. So if you are actually developing a molecule which will be having the size lesser than the pore of the lumen membrane, the infiltration rate of the molecule will be even much more higher. So the retention factor of the molecule into the specific compartment of the body will definitely be less, and the reachability of the molecule due to the surface engineering by applying certain kind of linker will be quite high. So the time taken by the molecule in different compartment, as it is mentioned, like probably for certain drug in some of the very good pharmacological books, where probably the retention type of drug is 4 hours, but by modifying the property you can even make the retention time far higher or far lesser, depending upon the kind of surface engineering do. As long as the matter of pharmacodynamic is concerned, it is the matter where the concentration of the drug will play a major role. So if by utilizing the nanotechnology you are providing the exact concentration to the site where the response is being given, then definitely it will stop the hepatic first pass effect, gut wall enzyme based effect, and through minimum dose the maximum effect can be observed. So if it is only PD and PK, then definitely these are certain factor which we have to consider. As long as the ADMET properties are concerned for certain molecule, which are actually considered as a nascent property of the molecule, whether it come to the toxicity, because it is concentration uh,
Yes.
based thing. So adsorption, distribution, metabolism, or even the excretion of that particular thing, can definitely be modified by using different kind of approaches. whether you want to, like if I say distribution, so you applied a linker where you want to distribute a molecule into the stomach only by using different floating technique. You may even go for GRDDS technique, or high and even low density approaches, where if you want to retain the molecule into the stomach, if you want to retain the molecule in the intestine, you may even use different kind of linker methods, mucoadhesive polymers, polyacrylic acid based polymers, so that you can even increase the distribution of a specific molecule for the specific time. So modification in ADME can also be done if it is done smartly, by considering all the biological, biopharmaceutical factor and physiochemical factor, and then we can develop definitely a promising molecule by utilizing these kind of advanced technique. This is, I think, first time I'm talking to the Dr. Harshita Tiwari, though I've seen her earlier also. So I think the whole responsibility of DCRC are there on you both, shoulders of you both, and I hope that you people will do miraculous stuff by utilizing this platform. So heads off to your effort, even after doing PhD. Along with the PhD, I should say, for Ashutosh, then you people are really doing great. So thank you very much for giving this opportunity. And if you have any further question, you may definitely shoot it out. Thank you very much.
Thank you so much, sir.
Thank you.
And
Thank you very
ma'am,
much.
do you have any query?
No, I would just like to thank Dr. Ashwani, sir. So, sir, thank you for your kind and encouraging words. And I appreciate the answer you gave. It's really very comprehensive yet very informative for me. So, thank you very much. I'm really grateful for your answer. Thank
Thank you
you,
so
sir.
much, ma'am. I would like to welcome Nilima Goswami, ma'am, for vote of thanks to Dr. Ashwani, sir.
I'm audible.
Yes, ma'am, you are audible.
Uh, Ashwani, are you there? Yes, you are there.
Yes, ma'am. Yes, ma'am.
हाँ, काफी दिनों बाद मैं देख रही हूँ अश्वनी, अच्छा लग रहा है। और बहुत दिनों बाद ऑनलाइन प्लेटफॉर्म पे आए हैं हम लोग। तो थोड़ा सा अभी एक्चुअली ड्यूरिंग कोविड हम लोग बहुत फेमिलियर थे, अब ऑफलाइन हो चुके हैं, तो दोबारा से ऑनलाइन आना थोड़ा सा अभी मुश्किल लग रहा है, क्योंकि जूम पे मैं बहुत दिनों के बाद आई हूँ, तो मैं के फीचर्स थोड़ा सा भूल चुकी हूँ। तो अभी ना वो समझ में नहीं आ रहा है कि कब म्यूट हो जाते हैं, कभी अनम्यूट हो जाते हैं। और अश्वनी, आप ये जो अभी शुक्ला जी ने जो ये शुरू किया है सॉर्ट ऑफ मूक टाइप नैनो टेक्नोलॉजी का, अच्छा है। एक्चुअली क्या है कि ये जो टेक्नोलॉजीज हैं ये थोड़ी हाइपोथेटिकल हैं, जिनको ना जो चीज हमारे सामने नहीं होती उसको हम एज्यूम करके हम बातें करते हैं। क्योंकि आप अगर आप डिसोल्यूशन देख रहे हैं या टैबलेट का हम कुछ देख रहे हैं तो ठीक है, लेकिन अश्वनी ने बहुत अच्छा बताया है उसको, और मुझे ऐसा लग रहा है कि सब कुछ तो अश्वनी बता चुका है, अब मैं क्या बताऊंगी। तो
Ma'am, it's not like that. You
तो
are
लेकिन
even a ocean of the.
ये टॉपिक इतना वास्ट है, बट थैंक यू अश्वनी, आपने ना मेरा काम आसान कर दिया है, क्योंकि आप पूरा बता चुके हो, तो मुझे ज्यादा उसको इलेबोरेट करने की जरूरत नहीं है। मैं जस्ट उसको थोड़ा सा अब कवर अप करने की कोशिश करूंगी। और बहुत अच्छा लगा इस प्लेटफॉर्म पे आके, और बहुत दिनों बाद आप लोगों से मिलके। थैंक यू, और मुझे बुलाने के लिए भी थैंक यू।
Thank you so much, ma'am. And I would like to request all the participants, if you have any query, you can unmute yourself, or you can put your queries in the chat box. So I think, sir, everyone is satisfied.
मेरी आवाज ना जाए इधर से, सिर्फ उनकी आवाज सुनाई दे मुझे।
So I think, sir, there is no query from the participants. Maybe in after the session. So yes, sir.
मतलब म्यूट
So,
म्यूट करने
so thank you so much, sir, for joining us. Please stay with us, and we have another lecture from Goswami ma'am. So thank you so much, ma'am. Sorry, sir. So I would like to.
Before moving ahead, I would like to say one thing to Professor Nilima Goswami, ma'am.
Yes,
That
sir.
thanks for her such a motivational word, and for the word of appreciation. She is the one who personally I consider as a ocean of the knowledge, and I should say that whatever I say, she is the one who has been inspiring, probably from the day when she joined the department, even before that, because in various platforms we have heard ma'am, and she is not only symbolizing an ideal faculty of a remarkable university in Madhya Pradesh, but she is the one who is actually bearing the responsibility to influence thousands and millions of the pharmacists with the energy she has, with the knowledge she has, and with the simplicity she carries most of the time whenever she do any kind of gesture. So thank you very much. It's even much more pleasurable for me to get these word of appreciation from my side, and I would definitely be in touch with you so that I may get some more word of appreciation from your side. Thank you
थैंक
once.
यू अश्वनी, थैंक यू। एक्चुअली हम दोनों के गुरु सेम है ना, तो इस वजह से सेम इन्फ्लुएंस है तुम्हारे ऊपर भी और हमारे ऊपर भी।
Definitely, ma'am. But in that case, also, you have got much more to share. I'm still in the pipeline of learning. Thank you very much, ma'am.
ओके, थैंक यू
Thank you
अश्वनी।
so much, sir. Thank you so much, ma'am.